All right. So one more minute. Okay. So today we're going to do bioenergetics. And more importantly, in bioenergetics, I want to focus completely on photosynthesis because in the first class, the very first class, we did all of this. We did ATP, we did aerobic respiration, glycolysis, um, energy currency, fermentation was the anaerobic method. And I also explained this on the first class, and I sent the YouTube lecture is on my YouTube channel for anaerobic respiration. You can check that out. So yeah, today I'm going to focus on photosynthesis. So photosynthesis, the very famous equation, which is 6 carbon dioxide reacting with 6 molecules of water, would give me C6H12O6. This right here is glucose plus I'll get 6 molecules of oxygen. Now, in this is for photosynthesis. For what do you call that? Respiration, right? For respiration, it's the opposite. So it's not this way, it's this way. So oxygen, 6 molecules of oxygen reacting with glucose would make you water and carbon dioxide. This is respiration. But but for uh, for photosynthesis, it's like this. So carbon dioxide is being used in photosynthesis, and in respiration, you use oxygen. So this is the photosynthesis equation. You have to memorize this. And photosynthesis basically has two reactions involved. The first one is categorized into the light dependent reaction, and the second one is light independent reaction. The first one is light dependent, and in this reaction, you will use light energy. But in the second, you won't use light energy, so it's independent of light. So the summary, this is basically the summaries of the first and second reactions. So light dependent reaction, right? You first trap light energy by photosynthetic pigments in chloroplast. You in your lower classes, you've heard of chlorophyll, right? Chlorophyll. Chlorophyll is basically a pigment which would absorb light energy. So the role of chlorophyll is to trap light energy, and energy produced because of this trapping of light energy, you make ATP and NADH, or basically reduced NADP. And this energy, right, this ATP is then transferred to the next reaction, which is the light independent reaction. And in this reaction, right, this is also known as the Kelvin cycle. And in this reaction, you will make molecules. So you would make glucose, you will make maltose, you'll make all of that stuff needed for growth. So you'll make all of the main nutrients for the plant, and the energy from the light dependent reaction is used for fixation of carbon dioxide to produce complex ions. So in this reaction, right, the light independent reaction, this is the reaction where you'll use the carbon dioxide that you see in this reaction. So carbon dioxide is used in the second reaction, the light independent reaction. All right. So let's start with chloroplast pigments. Now, what does chloroplast pigments do? Uh, they trap light energy, as I said, and they're found in the thylakoid membrane of the chloroplast. Remember, all of the photosynthesis is happening inside the chloroplast. So the pigments, right, the chloroplast pigments are also found in the chloroplast. And where is it found in the chloroplast? In the thylakoid membrane. The thylakoid membrane. Remember this. I'll explain more for about the anatomy of chloroplast. For now, just know that uh, pigments are found in the thylakoid membrane of the chloroplast, and uh, the pigments, right, are arranged in light harvesting clusters called photosystems. So these pigments, they are in many, they are bulged together basically in many so they are combined together, and these combinations, right, are called photosystems. So all of the pigments, they come in one place. Many pigments are concentrated in one place, and that's called photosystems. And different pigments absorb different wavelength of light. Uh, and remember that there are two groups of photosynthetic pigments. There are two types or two groups, as I said, of pigments. So these groups, right, when many pigments, they combine together, they form a group. And there are two types of groups you have to know. The first one is the very famous one. It's called the chlorophyll. And there are two types of chlorophylls, chlorophyll A and chlorophyll B. Whereas the second type of group is called the carotenoids, the carotenoid. And there is B carotene, and the second type of carotenoid is xanthophyll. Xanthophile. Yeah, like this. And yeah, so chlorophyll is of two types, A and B. Chlorophyll A, chlorophyll B. The second one, the carotenoid, is also of two types, the B carotene and xanthophyll. So chlorophyll is the only primary pigment. Chlorophyll A is the only primary pigment. The rest of the pigments are accessory pigments. So I want to focus on chlorophyll. Chlorophyll is of two types, A and B, right? This chlorophyll, chlorophyll A, is of two types. Chlorophyll A is called the primary pigment. And the rest of the pigments, right? The carotenoid, which is of two types as well. There is the B carotene, and there is the xanthophyll, right? And all of these, remember, all of these three pigments are known as the accessory pigment. Accessory pigment. Why is that? Because they would not be used a lot in the photosynthesis. This is the most important pigment, chlorophyll A. I'll get to what primary and accessory is, but for now, just know this. Moving on, the pigments, right, are arranged in light harvesting clusters called photosystems. As I said, these pigments are arranged in different groups, and this group is known as photosystem. A photosystem is basically different pigments in one group. So a primary pigment, which is the chlorophyll A, absorbs light and acts as the reaction center in light-dependent reaction of photosynthesis. So this chlorophyll A is the primary pigment. And why is it called the primary? The primary pigment basically absorbs light energy and acts as a reaction center in the light-dependent reaction. Whereas the accessory pigments, they surround the primary pigment. Accessory pigments may absorb different wavelength of light, and light energy is then passed to the primary pigment. So the role of accessory pigment is also to absorb light energy, but this light energy, right, this light energy would be transferred to the primary pigments so that electrons can get excited here. I'll get to that part. But for now, remember, accessory pigments, they transfer light energy to primary pigments. All right. And there are two types of photosystems you have to know. Photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2. Yeah. And this, I don't think you have to know this because this is the wavelengths. It's not written in the syllabus. So ignore this part. So let's focus on the light dependent and independent reactions now. So let me focus here. We don't need to know the anatomy of photosystems. Hanan, yes, you don't need to know the anatomy of photosystems. What you need to know is the anatomy of chloroplast and where the photosystems are stored. And I'll talk about that in the last 10 minutes. But yeah, so light energy from the sun, right? Light energy from the sun is absorbed by the light dependent reaction. The light is absorbed in the light dependent reaction. And this light dependent reaction also involves water. Let me go back to the first equation, right? In this equation, carbon dioxide and water, right? This water is being used in the light dependent reaction, whereas this carbon dioxide is used in light independent reaction. So remember, light dependent is where water will be used. So yeah, let me go back. So light dependent reaction is where water is used. The energy that is being used in the light dependent reaction is in the form of ATP. So in this reaction, right, ATP is made, and this ATP is then transferred to the light independent reaction. And here in the light independent reaction, you'll use that ATP and make different kind of nutrients. So glucose, maltose, all of that stuff. Sucrose, yeah, all of that stuff. So let's start with the light dependent reaction first. The light dependent reaction happens in the thylakoid of the chloroplast, the thylakoid. And its job is to trap energy and use that light energy to excite electrons in the chlorophyll. So this light energy is used to excite electrons. This is known as photoactivation. Photoactivation is when you use the light energy to excite electrons for the electron transmission chain. Will you share these notes today? If you want, I can, but yeah. So electrons are excited. This is known as photoactivation, and this is it's excited for the synthesis of ATP and NADPH. And also in this light dependent reaction, water would split into protons plus electrons. So there are two pathways of light dependent reaction. The first one is the non-cyclic photophosphorylation, and the second one is cyclic photophosphorylation. So light dependent reaction has two types, non-cyclic and cyclic. Starting with the non-cyclic photophosphorylation. So the very first step in the light dependent reaction is there are two types, right? The non-cyclic. So in non-cyclic, the first step is photoactivation. What was photoactivation? Photoactivation was basically light energy is used to excite electrons. So light energy is absorbed by the photosystems. Remember what was photosystems? There are two types, photosystem 1 and photosystem 2. So both of these photosystems use light energy, they absorb light energy, and this energy is passed to the primary pigment at the reaction center. So all of this light energy is transferred to the primary pigment. And in this primary pigment, electrons are excited to a higher energy level. So this primary pigment is where electrons get excited. And then after electrons are excited, the electrons are emitted from the reaction center or the primary pigment. Basically, the second name for primary pigment is reaction center. Reaction center is because electrons are excited and emitted from the primary pigment. So when the electrons are emitted, they are captured by electron acceptors. And what these electron acceptors do is they transport these electrons to the electron transmission chain for ATP synthesis. So electrons are passed along the electron carrier of the electron transmission chain. Remember, it's basically like respiration in mitochondria. So electrons are first excited, then this happens at the reaction center or the primary pigment. And then step 2, they are emitted and these emitted electrons, the excited electrons, are accepted by electron carriers. And then these electron carriers, they take the electrons to the electron transmission chain. This electron transmission chain is also found in mitochondria. And in this electron transmission chain, right, this electron transmission chain has ATP synthase, and ATP synthase will synthesize ATP. All right, so this was basically the summary of the first reaction, the first step, the light dependent reaction. This was basically the summary. So where was I? So electron transmission chain is where the electrons will go, and electrons released energy to produce ATP using chemosis. What was chemosis? I talked about this in the class of mitochondria. Chemosis is when the electrons and protons are used to synthesize ATP. And this chemosis happens due to ATP synthase. And ATP synthase is, remember, it's a protein that synthesizes ATP. So ATP made, right? When ATP is made in the light dependent reaction, this ATP is passed to the light independent reaction. And electrons are passed to photosystem 1. So remember, this electrons, right? These electrons, these excited electrons were excited in the primary pigment, right? These electrons were excited in the primary pigment, and then the energy from these electrons are used to make ATP. And when the energy is used, these electrons would not be excited anymore. So the electrons are not lost. These electrons are simply de-energized, right, when you make ATP. And these electrons then go back to the photosystem, the photosystem 1. Photosystem 1 is basically the primary pigment. And here, this electron would get light energy. This would get light energy. The electrons get light energy, they get excited, and they go back to the electron transmission chain and again synthesize ATP. So this complete cycle, right? This cycle where electrons are energized and de-energized, this is because of this, it's called cyclic photophosphorylation. This reaction is known as cyclic because of these electrons. So there is also photolysis of water. Why is water photolyzed? Photolysis is basically when water splits into two protons plus half oxygen plus two electrons. Why does photolysis happen? Photolysis is when the water splits. This is because you want to remove electrons from the water so that these electrons can be used for making ATP. So basically, water is used because you want to get more electrons for synthesizing ATP. Yeah. And electrons are replaced by these water molecules. So electrons, there are two ways of electrons getting replaced. The first one, let me zoom in, is through photosystem 2. Electrons from photolysis of water, right? This happens in cyclic photophosphorylation. Remember, water splitting, when water becomes electrons, this is happening in cyclic photophosphorylation. And you get electrons when you split water. That's why you need water. And these electrons would be then energized. Yeah. And the second way you replace electrons is when electrons, the de-energized electrons, become energized when it goes to the primary pigment or the photosystem 1 or photosystem 2. So photosystem 1, this was the first method where electrons are gained from photolysis of water. This is happening in photosystem 2. In photosystem 1, right, electrons are accepted by the NADP. NADP is reduced to NADPH. NADP is basically an electron carrier. or a proton carrier as well. So the electron is taken by NADP, and then it goes to the photosystem where it gets energized. These are the ways electrons get replaced or recycled. Yeah. So this was cyclic photophos. This right here, all of this is non-cyclic photophosphorylation. In cyclic photophosphorylation, uh, cyclic photophosphorylation only involves photosystem 1. Now, this cycle, right? This cycle where electrons get energized and de-energized, this is also happening in the cyclic photophosphorylation. So what's the difference between cyclic and non-cyclic? In cyclic photophosphorylation, electrons are recycled. And in non-cyclic, electrons are also recycled as well by electron carriers. Basically, what I explained, primary pigments make the electrons energized, then they become de-energized in making ATP. The de-energized electrons go back to the primary pigments, get energized. All of this cycle, it happens in both cyclic and non-cyclic. And photolysis of water splitting, right? H2O becoming electrons. This happens only in non-cyclic. So non-cyclic photophosphorylation, you split water into electrons. In cyclic, this does not happen. This is the main difference in cyclic and non-cyclic. So in cyclic, only electrons are recycled. And in cyclic, right? In cyclic, you only involve photosystem 1. In non-cyclic phosphorylation, you have both the photosystem 1 and photosystem 2. Now, in photosystem 1, electrons are recycled. How I explained it again and again, the energizing and de-energizing of electrons. Energizing happens in photosystem 1. Electrons are energized in photosystem 1. And in photosystem 2, right, you are photolysing water. So the photolysis of water happens in photosystem 2, where water splits into electrons. This is happening in photosystem 2. But remember, cyclic only involves photosystem 1. So this was cyclic photophosphorylation. It only involves photosystem 1. Everything else is the same. So electrons are excited in the chlorophyll A and then captured by electron acceptors. Basically, electrons then pass along the electron transmission chain. Energy is released by the electrons. ATP is synthesized by chemosis, and electrons return to the photosystems where it gets re-energized. Yeah. So there is also, after ATP is made, it is transferred to the light-independent reaction. This is where the light-independent reaction takes place. It's also known as the Kelvin cycle. And this happens, light-independent reaction happens in the stroma of the chloroplast. And this does not need light energy. So that's why it's called light-independent. And basically, there are 3 steps of light-independent reaction. The first step is the fixation of CO2. Then comes reduction, and then comes regeneration. In regeneration, the last step of light-independent reaction, regeneration, is when you make the nutrients. The important nutrients are made in regeneration. So first step is fixation of CO2. Carbon dioxide, right, mixes with a molecule called ribulose biphosphate. And when these two mix, right, when carbon dioxide mixes with ribulose biphosphate, you get 2 molecules of glycerate 3-phosphate. This reaction where carbon dioxide mixes with ribulose biphosphate to make glycerate 3-phosphate, remember, two molecules is made. This reaction is catalyzed by an enzyme called Rubisco. Rubisco is the short form. You have to know both the names, the short and the long. Basically, Rubisco is ribulose biphosphate carboxylase oxygenase. You have to memorize this name, ribulose biphosphate carboxylase oxygenase. This entire thing is an enzyme. So this is catalyzed by this enzyme right here, this reaction. So what after this, right, this step is known as fixation of CO2. After fixation comes reduction. Reduction is when this 2-glycerate 3-phosphate is reduced to something called triose phosphate. So this requires, remember this step, reduction, this is reduction. Reduction requires ATP and NADP. So reduction is when glycerate 3-phosphate becomes triose phosphate. And remember, since there are two molecules of glycerate 3-phosphate, you'll make two molecules of triose phosphate. So after you make triose phosphate, right, you will, this was reduction. After making triose phosphate comes the last step. It's called regeneration. So what is regeneration? Regeneration is basically when this triose phosphate, TP is basically triose phosphate, 1/6, right? There is 1/6 of this molecule is converted to carbohydrates, basically sugars. So 1/6 is converted into nutrients. What types of nutrients? You will make sucrose, amino acids, and lipids. And the rest of this triose phosphate, the rest of 5/6 of this triose phosphate is used to regenerate ribulose biphosphate. So 5/6 of this triose phosphate would be used to regenerate the ribulose biphosphate, which will be used in the very first step. Ribulose biphosphate comes from right here. So remember, 1/6 is used to make nutrients, 5/6 is used to make ribulose biphosphate. So this was regeneration. And remember, this step, regeneration also uses ATP. So you need ATP for regeneration and reduction. Yeah. So yeah, this was light independent reaction. So the main job of light independent reaction is to make all of this nutrients. Only 1/6 is used to make the nutrients. Triose phosphate, 2-triose phosphate, 1/6 of this is used to make the nutrients. So this is why the light independent happens. Now, I want to explain the function and the anatomy of chloroplast. So basically, this is the chloroplast. This is the chloroplast, and the chloroplast has it has two main things. So it has something called the grana and something called the stroma. So chloroplast is the main photosynthetic organelle. It's where light energy is converted into ATP, and you also make nutrients. And it has two phospholipid membranes. And remember that thylakoid, right? Thylakoid is this right here. So what is thylakoid? Thylakoid are these boxes right here. And when these thylakoid membranes, they stack on each other. So basically this is one thylakoid. When a lot of these, they stack on each other, they make something called the grana. This is the grana right here. All of this is the grana. Now, what is present in the grana or the thylakoid membranes? It basically, grana has large surface area because a lot of thylakoid membranes, they stack on each other. And the grana holds the pigments, enzymes, electron carriers needed for light-dependent reaction. And it also holds ATP synthase needed for ATP synthesis. And there is also large number of pigments to absorb light. And yeah, so all of the light dependent reaction is happening in the grana or the thylakoid. So remember, all of the light dependent stuff that is needed, basically all of this ATP synthase, enzymes, pigments, all of the stuff you need to make ATP is present inside the grana. And the second type of organelle in the chloroplast is called the stroma. Where is the stroma? The stroma is this empty space right here. It's basically the cytoplasm. It's like the cytoplasm. So the stroma is the site for light independent reaction of the Kelvin cycle. So stroma is the place where light independent happens. And you have in this stroma, you have the enzymes needed for Kelvin cycles. You have sugars, organic acids present as well. So the stroma is where light independent happens, and because of this, you will have the enzymes, the ribulose biphosphate, all of that would be present in the stroma. So this stroma, right? I told you that it acts like the cytoplasm. So the stroma also has 70S ribosomes, loop of DNA, lipid droplets, starch grains, all of that stuff. It also has ribosomes to produce chloroplast proteins. So stroma has a lot of the other stuff like the cytoplasm has. So basically you'll have the ribosomes, the DNA, the loop of DNA would be in the stroma, starch grains, chloroplast proteins like ATP synthase. You need ribosomes and DNA to make the proteins. So all of that would be present in the stroma. Yeah. So is there anything else? No, there isn't. So I want to focus on this question. If you can solve this question, basically you've understood all of the topics that I explained. This is from 2017, I think. 2014, I think. Yeah. You have like 2 minutes to solve this. Let's wait 1 more minute and then we'll solve the question. All right. So I don't think I'll get any answers. So no answers. All right. So let's solve this. Which one of the following is a correct outline of some main events in the photosynthesis of a healthy whey plant? So they're basically asking what would happen in photosynthesis. Let's go over all of these steps. Carbon dioxide combines with an acceptor compound right? This happens. This is known as fixation, where carbon dioxide mixes with ribulose biphosphate and breaks into, so this compound would break into two, and each is reduced by hydrogen split from water by light. Yeah, this is right, because remember that this compound, when this is made, this would be reduced, and hydrogen split, right? Hydrogen split, remember the hydrogen split, this is known as NADP, reduced NADPH, reduced NADP, or NADPH. So this NADPH, remember, it's a hydrogen acceptor, hydrogen acceptor. So this carries hydrogen, the NADP carries hydrogen. I explained right here as well that you will need NADP in reduction, NADPH in reduction. So yeah, hydrogen is used in reduction. So A seems right. Let's go to B. Light splits from water. Light splits water, and the resulting combines with a compound which has reacted with carbon dioxide. Uh, Light does not split water. You don't need light to split water. So this is basically wrong. And also the hydroxyl group. What is hydroxyl group? Hydroxyl is the OH group. You don't have OH group, right? You don't use OH group anywhere in photosynthesis. So yeah, this is wrong. C says that in the presence of light, oxygen reacts with a carbon hydrate to produce water and carbon dioxide. Remember that the reactants in photosynthesis are carbon dioxide, not oxygen. So oxygen does not react with carbon dioxide in photosynthesis. This right here, all of this happens in respiration. So yeah, if this was opposite, if they said carbon dioxide reacts with water and make carbohydrates plus oxygen, then yes, this would be right. But in this case, this is wrong, because this is basically respiration, not photosynthesis. And finally, light splits carbon dioxide, and the resulting carbon then combines with oxygen and hydrogen obtained from water. What? No, this is wrong. Carbon dioxide does not split. Water splits. And E finally says that light joins carbon dioxide to an acceptor compound, which is then reduced by hydrogen obtained from water. Light does not join the carbon dioxide with ribulose biphosphate, because remember, this step is happening because of light independent reaction. This step is light independent reaction. You don't need light energy to cause this reaction. So this is also wrong. So yeah, the answer to this question is A. Is this clear? All right. Thank you for this class. See you tomorrow. Bye.